Greetings, my name is Elise Cartagena. I'm an undergrad student from Dr. Velasquez Research Lab, and I will be presenting my project, which is titled Ethanol Modulation of Surface Expression of the BK channel is isoform specific. So when we talk about tolerance, we can define it in a higher level as a low response to a drug after a person being exposed to it to a higher extent or a prolonged exposure. When we talk about molecular tolerance to acute ethanol exposure, we can relate this to BK channels or big potassium channels. Because these channels, they have been proven to play a key role on the development of molecular tolerance to the acute exposure to ethanol. Why is this? They show a high response to this exposure. This channel is different in isoforms, and they are composed of two subunits, being them alpha and beta subunits, and they respond differently due to the difference in the alpha subunit being them different in 58 amino acids, differentiating them in specificity of these different isoforms. So the gap in our knowledge would be the following. Even though we know that there are changes and these changes are dependent on a timely matter, and they are also dependent on the type of isoform that we are working with. There is still an unknown knowledge or a gap in the knowledge on how this ethanol exposure is going to affect the protein levels, and when we talk about a specificity of this isoform and how these protein levels change after such exposure. How does the expression of this BK channel, which is isoform specific, is affected by this exposure to ethanol? Well, that's the question that we are asking here in our project. So we hypothesize that the redistribution of these BK channels are going to be a important aspect for the development of molecular tolerance to the acute exposure to ethanol. The methods that we used in this project were cell cultures. We did cultures for human embryonic kidney cells, or HEC293 cells as they are more commonly known. And we also did hippocampal neuronal cell cultures. We did immunofluorescence protocol also, and we did turf imaging and analysis. We did Western blot protocols to measure different protein levels on a cellular level in HEC293 or human embryonic kidney cells. And we also perform electrophysiology to study the kinetics of the channel in their different isoforms. So the mechanism and the components that we propose for the molecular tolerance to ethanol exposure is going to be the following. We first have the potentiation, or this channel that is being responsive. This channel is going to be clustered across the membrane, and these clusters are going to be distributed in the surface. And we also have expression of the channel inside the cell. After 24 hours or chronic exposure to ethanol, we see desensitization of the channel. As we can see here in our graph, we see that the channel stops responding. We see declustering of the channel, and we can also see a redistribution of the channel across the membrane and internalization of the channel. For the different kinetics of the different isoforms, we have insertlets, which is another name for zero isoform, and we have Streck's isoform. For zero isoform, we see here that after 3 minutes of exposure of ethanol, we see that potentiation of the channel, and then as time passes by, we see that that decrease, that desensitization as time passes. On the contrary, and here we see the specificity of the response in the different isoforms. We see that Streck's isoform is not going to be responsive to the presence of ethanol in the cell. For the zero isoform, we see here the membrane expression, and as a result, we have that the expression of the channel it's going to be lowered after 10 minutes and 24 hours of exposure, as well as declustering of the channels. On the quantified data, as we see here in the graph, we can see that for the fluorescence of the intensity of the membrane, we see a lowering after 10 minutes and a significant decrease after 24 hours of exposure. And after that, we see here the mean intensity of the clusters. We see here a significant decrease after 10 minutes and 24 hours. This is going to be, it suggests that it's due to that desensitization of the channel, and that mechanism proposed that the channel is going to be internalized after that desensitization and redistribution. Here, we did Western blot. We performed a Western blot for the BK isoform because we needed to see where that channel went. And after 24 hours, we performed Western blot protocols, and we see here that for the expression of the level of protein normalized to beta-actin, we see here that there isn't any changes in levels of protein of that channel in the cell. On the contrary, for the isoform of Streck's, we see that we don't see any decrease in that membrane intensity as well for the threshold, and these can support the data that suggests that this channel is not being responsive to the presence of ethanol in the cell. We see here that after 10 minutes and after 24 hours, the channel still behaves the same way, as well as we see clustering that is not being redistributed across the membrane or nor internalized in the cell. And then we went ahead and did the same protocol, but this time for endogenous surface expression of this population of the BK channel. We did our turf imaging here. As you can see, the BK channel staining, and because we're working here with endogenous population, we don't have a way of telling if this is going to be either Streck's or zero. So here we are working with all the isoforms that the neurons have to offer us to work with, and we can see here that after 10 minutes we see that there's an increase in membrane intensity, but after 6 hours that intensity decreases, and after 24 hours we see that it gets normalized with the control. As well as for the clustering, we see that it behaves the same way as the membrane intensity. After 10 minutes it goes higher in intensity, and then after 6 hours it decreases, and then it gets normalized again with the control. We measured the clustered intensity, and we also measured the membrane intensity for 6 hours of exposure, and we did this tagging that Streck's isoform with a GFP tag, and so we could know that and see that concentration for us to be able to quantify it. So we see that for the mean membrane intensity after 6 hours of exposure to acute ethanol, we see that there's an increase in concentration of these channels, as well for clustering. This helps or supports these mechanisms that the alcohol-sensitive isoform is going to be internalized, leaving us with a non-sensitive isoform expressed in the membrane of our cells. So in conclusion, the data suggests a timely dependent reorganization on the surface of the membranes, and it's going to be isoform specific for the BK channels and the exposure of ethanol upon them. And we can see this behavior in neuronal endogenous populations of the BK channels, and we can also see this in the embryonic kidney cells model. We also know that the effects of the expression and modulations of ethanol exposure in these channels is a relevant component on having a broader view on these mechanisms of development of molecular tolerance to the exposure of ethanol, and this can help us to trigger these mechanisms and then be able to prevent these development of molecular tolerance beforehand. And that's why it's so relevant to have a much larger view on this topic. Thank you so much for the attention, and I hope you enjoyed it.